Hello, everybody. So this is the fourth part of our vaccine series, and hope you enjoyed the TED Ed talk that was the third part. So let's take a look at Edward Jenner. 1796 essentially came up with the first safe and effective vaccine. So one of the interesting things about his story was that he himself never came down with smallpox. And they say that's because he was variolated against smallpox when he was young. The second thing is with his research, essentially what he did was use a less virulent relative, if you will, or similar virus, cowpox, and use the infectious materials from that to induce immunity that then gave people resistance to smallpox. And he did that through very clever and repeated studies. So he had used the son of his gardener, I believe his name was James Phipps, and he essentially inoculated him with the cow infectious material from the cowpox lesions. And then 2 months later, then he challenged essentially this boy's immune system by then exposing him to smallpox. He did that a few more times to essentially say, now he's resistant. If you take a look at the picture here on the right, 1901, you essentially see an unvaccinated boy with smallpox on the left and one that's vaccinated on the right. So when you think of pox, whether that's chicken pox, smallpox, cowpox, you're thinking about these skin lesions, which can be really, really uncomfortable. And of course, with smallpox, we know that it literally changed the history of the earth because of the number of people that died as a result. All right, so this is just a summary slide. If you like to see these things kind of written out in words, if you want to take notes from that, you can. A couple of things about the word vaccine. Vaccine comes from the Latin vaxa, which refers to cow, since vaccines were originally discovered from using cowpox. Okay, so the definition of vaccination, any immunity obtained by inoculation with selected antigens. So this is where we're going to go a little deeper with different kinds of vaccines and how we make them and how they work and some pros and cons of those vaccines. All right, so here's the general concept of vaccination. I don't believe this figure is in your textbook, but it's pretty helpful to take a look. So on the far left-hand side, we see the disease-causing microorganisms. So maybe this is Hep A, maybe this is measles or pertussis or COVID. So here's the disease-causing microorganism, and normally what would happen is that we get an infection from that microorganism, and then our immune system is going to launch a defense. So anytime you get a common cold or maybe norovirus or some other case of gastroenteritis, then you know generally what's going on with your immune system. You have some symptoms. The symptoms usually are indicative of various kinds of diseases that we might have picked up, and it's going to take a certain amount of time for us to recover. Sometimes we recover, but we have some inadequacies, so just not quite feeling the same for a while our body systems go back into homeostasis. So the far right-hand side, so for any particular disease, we can see there's kind of 4 outcomes. Certainly, mostly, this happens a lot of the time, we recover, right? So we recover, no big deal. Second thing that can happen is death. All right, so that is a big deal. We've succumbed to that particular microorganism. The third option there is disability. So when we think about polio, yeah, you can survive, but you can have lifelong physical disabilities because of it. And then lastly, we know that in any of these cases, particularly if we are surviving, even with a disability or complete recovery, that we have immune memory to that thing we were exposed to. In the second situation here, we see, okay, this is vaccination. So we don't acquire the whole microorganism as it's normally circulating in the population. Instead, we are exposed to some part of that. So we need to talk about vaccines in just a minute. But essentially with vaccination, you are exposed to the antigen for that particular microorganism. And without you even having to do anything or feel crappy or anything else, your immune response is launching. And so it's going through the various cascade of events and eventually will make a special type of B cell that cranks out memory cells, and those memory cells will make antibodies when or if and when you're exposed to the actual disease. So in this case, we're looking at a smiley face that you're going to fight it off without even knowing that you did, and are able to recover. So this figure shows us 4 ways to make a vaccine. And I have spent a couple of slides going over this for more details. But essentially, we can see far left-hand side, we have inactivated vaccines. So we're going to use a killed version to trigger the immune response. So we can kill that particular microorganism by chemicals, radiation, heat, or some other agent that essentially is going to kill the virus but not change its antigenicity. So that means the antigens are still going to be there, and so we don't get the actual virus, but instead we get the antigen part of that virus. So it has been killed. It's not going to be infectious, okay? So that is an inactivated vaccine, and this is considered a whole cell vaccine because we're essentially using the very organism that would make us sick, but we are inactivating it. Okay. Another whole cell vaccine is an attenuated virus or an attenuated vaccine. So essentially, we are going to weaken the virus, but it's still going to trigger an immune response. So we can modify the growth conditions in which we are growing this particular microorganism, or we can actually manipulate bacterial genes. So essentially, we have a weakened version of the virus, and we'll use that to trigger the immune response. On the right-hand side, then we essentially have subunits or subunit vaccines. The first one does say it's a subunit vaccine, so we're only going to use a portion of the virus. Usually we're thinking about spike proteins, so that is going to teach our immune system about the exposure of that to that particular microorganism. And then the last one, not the last one, also with subunit vaccines, we can make what we call toxoid vaccines. And so in this particular case, we are purifying the exotoxin or a toxin associated with the particular microorganism. And it's that toxin that makes us sick, not necessarily the actual organism getting into our body, but the toxin that it makes. So essentially, we are forming our vaccines that are going to trigger the antigen, or this toxin, or this toxoid is going to trigger our immune system to make antitoxins against that particular toxin. So we call those toxoid vaccines. And the newest, latest, greatest one here is the nucleic acid vaccines. So we're going to use actual virus DNA or RNA to enable our human cells to manufacture portions of a virus to trigger the immune response. And we'll come back to that in a little bit. Okay. Again, if you like to have some notes, here's some notes. I want to go into this a little bit differently in the next few slides. So if you want to take a look, you can take a look. Okay, so more details of types of vaccines. All right, so here are 2 types of whole organism vaccines, the killed cell or inactivated virus, and then the second one, the live attenuated cell or virus. So we talked about that, but essentially what we do is we use the whole organism to stimulate the immune system, but it's not going to cause disease, okay? So examples of killed cells or inactivated viruses include the flu vaccine. Most of the flu vaccines fall in this category. And then the second one there, the live attenuated cells or viruses, includes measles, mumps, rubella, and I have one more. I believe it's on the next, on another slide. But essentially, we are weakening that live virus so that it's not going to make us sick, but essentially it is going to it's going to act as the antigen that's going to prompt our immune system. Now, the thing about this particular one is that we actually have the living organisms in our body. Yes, they have been weakened, In the very rare situation, they could mutate, but it's very rare. But essentially, these vaccine microbes can multiply and boost our immune stimulation. So they are very, very, very good vaccines. Okay, this other category, the subunit, or which include toxoids, but also our antigenic, what they call antigenic vaccines. Essentially, what we're doing is we're taking a particular subunit or a particular antigen, and that's what we're using to induce the immune response in the host. So in the first one, you can see we're taking essentially spike proteins, injecting that, or doing the process to make the actual vaccine, but then using those surface antigens or spike proteins to initiate the immune system. So this is like hepatitis B vaccine. And the second one here, so we need to talk about this a little bit further, is essentially using messenger RNA, which is coding for the spike proteins. And we're putting that messenger RNA into our vaccine in a particular form. And then that is going to essentially be taken up by our human cells. And our human cells are going to make spike proteins. No, the messenger RNA is not enough to make essentially, for example, COVID. It's not going to be replicating in your body and making COVID from that. This messenger RNA is just simply coding for those spike proteins. The spike proteins on the outside of your cell are going to initiate an immune response so that we have antibodies against the spikes. Okay, so that's like the original Pfizer and Moderna COVID-19 vaccines. All right, in the third situation here, again, a subunit or antigenic component vaccine. They are taking surface antigens, and then they're making a complex with a protein to increase its antigenicity, which is essentially going to create a stronger immune response. So we see an example there. And then lastly, the Johnson & Johnson vaccine was using DNA or RNA, but essentially it was a harmless virus that was going to be used to initiate immune response against the surface protein of the particular virus. And so this could be COVID-19, it could be something else. So a little bit different than the Pfizer and Moderna versions. Okay, so let's take a little closer look at this messenger RNA vaccine technology and advantages to this. So essentially we've injected messenger RNA that's going to code for spike proteins. It's going to be taken up by your cell. So essentially the messenger RNA is packaged in a lipid vesicle, and we know that our cells are also covered in a lipid bilayer. So they're these lipid vesicles are very readily taken up inside our cells. And then our cells are going to make spike proteins that then is going to stimulate our immune system, in particular T cells and B cells. And they're going to make antibodies and, of course, a memory cell to those spike proteins. So it's there, ready to act on demand. So what we know about this is we can make these vaccines much quicker than all the other versions of our previous vaccines. They're highly effective, and there's usually very rare side effects, and if they are, they're harmless compared to the actual disease. Now, let's take a look at some pros and cons from the other types of viruses. So we'll start with the killed virus. So the killed virus, essentially, because it's been killed, it's not nearly as potent. So it takes a larger dose of that particular vaccine. We're going to need more boosters, essentially, to get our immune system to get a big enough immune response. And we know what happens over time is the immune system loses its, specificity, if you will, for killed viruses. The live attenuated viruses work really, really well because they're multiplying and they produce a pretty good infection inside of the host, but not one that's going to cause you to have any, side effects. But essentially then it's going to prompt a really long-lasting protection, fewer doses, doesn't have to be as strong, and less boosters. So it's very, very effective. It's just that the thing is we have to store them in a special facility. We know that these particular organisms can mutate and become virulent, potentially in their host, but it's very rare, but they're hard to make. All right, subunit vaccines work well for only some diseases, and the same thing for toxoid vaccines. So we will pick up and do